Okay, so we're back here and we're going to start looking at this. So this is, you remember I told you a little bit about this before. This was the idea of transposons, and we hopefully will get to those. If not, we'll surely get to them on Friday. They come at the end. And so I showed you that before. So these are the distinctions on genetic transfers are coming, not genotype or phenotype, any of the. We'll get to all of these and what that means. Again, auxotrophs mean, and we utilize them again when we get to something called the Ames test. I don't think it's a name that you have to know, OXO or proto, that are there. But every so often, if you see a test, not necessarily by me, if they have, this is, for instance, this is code for being resistant, like, let's say E. coli that's streptomycin resistant, or a strain of E. coli that requires the amino acid tryptophan to be able to grow. That's where those come into play. And at the very least, even if you down the road, if you're looking at research articles, that would be helpful because again, for many of you going on into these programs, research, at least being able to read and understand the research is important. This is the idea of mutations. We're making DNA. It's a very complicated molecule. There's a mutation rate that's consistent, okay, in living things. It could be as little as 1 in 10,000 or as much as 1 in a trillion. For eukaryotic cells, because of all those safeguards, it's a lot closer to the 1 in a billion to 1 in a trillion. For bacteria, because of the lack of those safeguards, it is a lot closer to the 1 in 10,000 to maybe 1 in a million. So it's proportionately lower by maybe a factor of 10 to the fifth, 10 to the sixth. Okay. We'll talk about what are called reversion or back mutations. That's that's an oddball. That's such a powerful mutagen. If it could mutate it backwards to being normal, imagine the effect of mutation. That's what's called the Ames test is based on. So again, you can. I think it's a misstatement to think the environment do cause mutation. If we have overabundance of sunlight and we didn't have adequate cloud cover, theoretically that would certainly cause different kinds of mutations. So without getting into uh, the evolutionary aspect of it, so we look at these. This is one of the reasons why for diseases and conditions, bacterial, that are difficult to treat because they mutate rapidly. Read that, particularly as tuberculosis. We'll use two kinds of antibiotics rather than one. This is the most common mutation. It's one of those simple substitutions in a base. Okay, these are DNA bases. We call it a point mutation. And effectively, this is the idea of the three different types. This is by far and away the most common. And so again, the three different types of these, whether they have no effect, silent, whether they alter an amino acid in a chain of a polypeptide or protein, that's a missense, or whether they encounter a premature stop code, that's a nonsense. Those are big trouble. So silent, missense, nonsense. And they're showing you here just arbitrarily because they always use this kind of an illustration. So the wild type, what you expect to find, would have this. Now, if you're only changing that last letter, it's still the same amino acid called cysteine. If you end up changing the letter here to slightly different, okay, now you get tryptophan. So that definitely changes the sense, maybe not alter it tremendously functionally, but will alter it. And here you have the stop code. And the stop code is big trouble. So it just gives you an idea how one isolated change could have no impact to having a major impact. It's just basically what they're talking about. So when you start talking about inactivating the gene, they call it, fancy name is a knockout. And we see a lot of these with us because most mutations come directly from reactive oxygen species, all of those toxic derivatives of oxygen, those free radicals that I mentioned quite a bit before, superoxide. That's one of the things that really causes a lot of mispairing. And down the road, even in the next chapter on viruses, we talk about it a little bit more. It's there to understand that if we don't have the means to deal with these toxic species, and bacteria don't, generally they have a greater tendency to mutate. So there's other ways that it can change as well. Remember, it's 3-letter patterns. So the worst ones are called frame shift mutations. Effectively, you can either add a letter or delete a letter. So imagine if you had a sentence that was all 3 letters, like you did when you're in first grade, the fat cat, something, sat, you know, that type of thing. And so 3-letter words. But if you added another letter in there, everything gets thrown off. So all you have to do is add like a TH, and all of a sudden you get the, and the next word would be cat, you'd have TFA. What's that? So all of a sudden it throws everything off what we call downstream. That's another way of describing a frame shift mutation. So if you add one, all of a sudden, rather than getting this pattern, you get a wild pattern, and sooner or later you're hitting a stop code. So anytime we see what they call addition or deletions in DNA, those are big errors. Point ones are fairly common and much more easily dealt with. This is the nugget about transposons. It's called a transposable element. It can literally add to a particular gene location. It can insert it. It can do different things. That we'll see an animation about it. It's not a big deal for us now. Down the road, when we talk about antibiotics, one of the things that is really the most troubling of all the infections that you'll continue to hear about still is MRSA, methicillin-resistant Staphylococcus aureus. It's not the only one. There's also methicillin-resistant Staphylococcus epidermidis called MRSA. One of the things we worry about with antibiotics is we're going to run out. So the drug of last resort that you'll hear about again when we talk about antibiotics is called vancomycin. So if you had a gram-positive infection, staph, strep, and it was out of hand, hey, give them vanco. Okay? You'll make it. Not anymore. Now we're seeing vancomycin resistance more frequently. We think that had a lot to do with a transposable element going from one bacteria to another. It went from a gut bacteria that was in the strep family called, as you will learn, called Enterococcus fecalis, and magically made its way to Staphylococcus. And these are the kinds of things why this is so relevant clinically. Not that you have to know about it yet, but in order for you to be interested in learning about these things, you have to understand the relevance and the importance, I think. Because now you get into these superbugs, and you're a big, that's why we're constantly looking for new antibiotics and ways to change them. The chemicals, understand a mutation is an error in DNA. Things that cause mutations, whether it's radiation, whether it's those transposons, whether it's different chemicals, okay, cause errors in DNA. They don't all mean terrible things. Okay. A mutagen is one thing. A carcinogen causes cancer. Down the road, you'll hear about something called a teratogen that causes birth defects. The terms are not mutually interchangeable. Okay. So the word, mutations are significant. Sometimes we have mutations that are beneficial. So you don't have to go into what these do. I don't care. A lot of these we use these in the laboratory for purposes to create things that are there. So we look for different kinds of things that we're trying to create. We call these induced mutations. Unfortunately, when you're exposed to noxious chemicals, when you're exposed to radiation or ultraviolet light excessively, that too can induce mutations. If you're an idiot like I am, or was, for 40 years and smoked cigarettes, you're going to be inducing a fair amount of mutations. So, for all of those reasons, when you induce a mutation, it increases the likelihood by a factor of 1,000 times. So in bacteria, they are roughly, they mutate somewhere between maybe 1 in 10,000 to maybe 1 in a million. So if you're increasing that by a rate, you're going to create bacteria that could mutate as frequently when you expose them to chemicals or antibiotics to a factor of 1 in 10. So understand, living systems are like that. You can't push on a living system and not expect it to push back in some way, shape, or form. It's just the nature of living systems. It's going to foster defense mechanisms, it's going to foster aberrations, and a lot of times the mutations that survive, by definition, are mutations that are harder to deal with because they're adapting themselves for survival. It's nothing more than, evolution, survival of the fittest. So, and that's something that medicine was not as cognizant of, how bacteria could do that. And it's not like they're reproducing one or two, they're producing, as you saw before with the growth curve, millions of them within a day. So there's lots of different things. Down the road, we're going to talk about these. These are things that we've made chemically called base analogs. Where do we use them? They are the highlight film for cancer chemotherapy. Chemotherapy is a misnomer. Every time, grandma's on chemotherapy. Well, I'm on chemotherapy. I take blood pressure medicines and arthritis medicines. Those are chemicals for a purpose. Cancer chemotherapy mostly incorporates these. These are base analogs. They put something in the base that is a look-alike drug, for lack of a better way. So they substitute effectively a methyl group for a bromine. Bromine is a halogen. Halogens are poisonous. We get too much of them. If you feed something a halogenated analog that's supposed to be for thymine, it's going to basically interrupt making DNA and RNA. So we use the original, this 2-aminopurine was one of the earliest cancer treatment drugs. We use more sophisticated ones today. If you know somebody, or if you've had to take a Valtrex for a cold sore, for a herpes infection, it works the same way. So there's nothing unusual under the sun about this. It's important to understand why these are where they are. We use base analogs. So when we get to antivirals, you're going to see a lot of base analogs because that's the principal way we treat them. We don't have anything naturally occurring that we can make. Typically, we can make something called interferon, which we make in our bodies, but there's no magical antibiotic. There's no living substance that can fight virus infections. Different ways you have to treat things. Intercalating agents, again, mostly in the laboratory. This is the one that's very relevant to us. These are thymine dimers. This is relevant to you. Okay? It's the whole idea is if you have sun exposure is funny because we've encountered a lot of problems with various kinds of sunblock agents over the years. So, and I'm not as up on it because I don't tan, I burn. Okay, I'm one of those. So, I mean, I like the beach except for the sand. I know that's hot.
It gets hot.
It gets hot, but some people are have got to be out there, but that's fine. You have to do it judiciously. This is the problem. Ultraviolet light, thymine dimers. Okay, it can cause a lot of problems. What it does is it creates this, and you might remember me mentioning it before. This is one of those methods of control things where it causes these two to bond together, and it creates that little irregularity in the chain of DNA, so it can't open or close. Okay, where X-rays, ionizing radiation, actually cause breaks in DNA indirectly because it makes more of those toxic derivatives of oxygen. Okay, and you saw this with the transposons before. So again, large amounts of either spontaneous or in the presence of a mutagen like radiation or ultraviolet light. Okay, so this is one of those things. For example, in humans, two genes associated with breast cancer, you've all heard of them, the BRCA genes, code for DNA repair enzymes. So if you have one of these, you're a higher risk for breast cancer. Breast cancer is horrible. We're talking about typically about 1 in 7 women have to deal with that. It's a nightmare. Okay. If you have both of them, your odds are substantially increased. Because remember, when I framed this to start, we have enzymes that can proofread and repair your errors in DNA. If you have errors in both of these, in two of these systems, the likelihood is tragic, potentially. That's why everybody wants to know somebody who they discovered a lump and they said, well, we're going to have to do it. And they found it was cancerous and they had those two genes. They elected maybe to have a double mastectomy. Most families have somebody in that category. Our family does. Okay. Mutations are rare because alterations in DNA generally are repaired before it's passed on to progeny, but it doesn't mean it's not there. So there's lots of different things, and we'll look at the repair mechanisms. I'll show you a bit about mismatch repair and different things that are associated with it. So regular DNA polymerase in bacteria sometimes corrects the wrong side. Like I went up and down the aisles like I always do and go, it's just likely to repair the good strand and make it into another bad strand. So there are different ways, and it's not necessarily effective. Mismatch repair sometimes fixes errors that DNA polymerase, but not terribly well without going into the dynamics about it. So I don't do a lot of these. And then these are different repairs. There's a thymine dimer. Interestingly, for us, for you and I, you went out, you're out too long and you got sunburn, and thankfully nothing bad happened to it, unless you were doing continuous exposure. We actually have actual light can repair some of these things. Okay, so we have a variety of mechanisms, not so much like bacteria with light repair, but we have a lot of different mechanisms inside our cells to repair it, as long as we don't have continuous or constant exposures. Give yourself enough time in between sessions, you're probably doing all right. Like if you like to go tanning, like I have a daughter. Every time I tell her, talk about this, my daughter, hey, what are you doing? Can't talk, I'm tanning. With her phone? She's one of those. Oh. So we have repairs cost energy. Bacteria don't make a lot of energy. That is the problem. These are lab things I'm not going to do. Okay, or plating I'm not going to do. Our bio majors and genetic majors do this. They use silk and plating techniques and things like that. These are things that if you wanted to make our micro lab more sophisticated, you could do so. But you will have media where you add things to. What'd you do this week in lab?
Dilution.
What?
Dilution.
You did dilutions. Okay, cool. So now this is the next note. This is what I talked about earlier. Cancer causers. So a mutagen that's also a carcinogen. Okay, most of them are mutagens. They cause errors in DNA. Understand, we don't like to test. Years ago, they would test these on animals. They're usually using animals that are relatively close to us, like dogs and cats. You don't even want to hear from me about dogs and cats. We did live experiment. We did something called vivisection. 50 years ago, doctors did this. It's horror. I have nightmares about it. Just to let you know. And as most of you know, except when my cats and dog are a pain, I love animals. My wife hasn't been home for 5 nights, so I have a buddy in bed who's white and a cat. It's like if I move into him, he goes, okay, got the picture. Turn the other way.
It's the dog's bed.
No, it's the cat owns
It's
the
cat's
house.
bed. You should be lucky
Cats
he lets
rule.
you sleep in it.
He's the cat. Cloud the cat. Okay, so mutagens have a very low frequency of what we call backwards mutations. So they typically can't reverse a mutant form back to normal. So only strong mutagens that are cancer causers do this. This is the basis of the Ames test. Everything the FDA, if they're going to give it to you, topically, orally, injectably, if you're going to go out and buy a hand cream or a lotion or anything else, a beauty product, it's had the Ames test. So it takes what's called a salmon. That's why I told you about auxotropes. It's a fancy name, which means this is a mutated form of salmonella that needs histidine. And what they do is they mix it. Now they have to take some human enzymes, so they sacrifice, it's a fancy word for kill, and take rat liver extract because it can convert it. Animal enzymes very often are required, and it mirrors what we would do. Okay, and it basically, if they find out by testing it that it has caused the salmonella to go back to normal, okay, prototropes means it's normal salmonella, then it has failed the Ames test. Okay, the Ames test, if it's a carcinogen, a mutagen, or that's strong enough to be a carcinogen, it has been able to reverse or backwards mutate the salmonella to normal. That's important because that's the kind of thing that they're going to ask you about in an NCLEX or a licensure exam. So I have a couple of questions on my test. Does the Ames test use salmonella? That you should be aware of that. Is it there to make sure things were safe? What kind of mutation causes a backwards mutation? Those are the test questions. Something along those lines. I'm giving them away for free. Tax deductible contributions are accepted. Okay. Now we get into this. There's 4 different animations I focus on, and I explain each one. I'm hopeful it will play without me having to tinker with it. They are called transformation, transduction, conjugation. The one you don't see here called transposition. Typically, we start with transformation. Okay. So these are what are known as horizontal or lateral gene transfers. How genetic material from one bacteria, even if it dies, can go to another bacteria and change it. Transformation and transduction do not require contact. The bacteria can just enter another bacteria, which does happen. And in order of frequency, this is the rarest, this is the next rarest, this is the most common. This is where, and these are depicting little viruses who have a fancy name called bacteriophage. Viruses can infect all living things, including bacteria and archaea. They do not infect us, so they do not impact us directly. But they can pass properties on to other bacteria, sometimes which are a problem. You'll see that down the road. And this, by far and away, the most common. This is the one changing a population from Staph aureus to MRSA overnight, called conjugation. The think contact transfer. It utilizes the F or fertility pilus or pilus, however you want to say it, and it's a very specific mechanism. Okay, we're going to look at origin of replication. I'll explain homologous recombination to you. It's a very interesting process. So before I get there, I'll sort of frame it a little bit. So we start with transformation. So bacteria, remember the growth curve. They're constantly living and they're constantly dying. When a bacteria dies, all that DNA kind of explodes out. When it explodes out, it starts to fragment before it deteriorates into its whatever its components are. So you have little short strands of maybe 20 to 50 genes floating around in the environment. So you have these little strands, and bacteria are moving like anything else that's living, and they will be contacted by that DNA. If it strikes them, this is why it's so rare, it has to strike them in the exact spot where there's a receptor. They have to be what's called, and here's the name, competent. They have to be in a state where they're able to accept. That's what competence means in this concept. We can do this in the lab by warming them and chilling them and exposing them to calcium salts. So there's nothing, this is done in the lab all the time. Some accept only DNA from the same species. Some will take it from anybody. Okay? And some of them are always competent, some are not. And it doesn't pay for me to show you this so much as to show you the illustration. But first, why this is so important. 1920. Nobody knew other than that DNA was important. Nobody had, there was way, this was 30 years more or less before we understood the molecule itself, before the Watson and Crick model, Nobel Prize winning work was developed. No idea. This doctor named Griffith, and later, about 40 years later, we found out he didn't know it was DNA or protein. 40 years later, they were able to tag it radioactively to tell you it was DNA. So he made observations. Strep pneumonia, the most common cause of bacterial pneumonia. Okay, it's only deadly when it's got a capsule. One of the things. We injected that into the test animal, in this case, a mouse. No, it's not good for us. If you took the non-encapsulated strain, because they look different on the plates in the lab. That mouse is fine. Then he took heat of the non-encapsulated cells, and interestingly, he killed the encapsulated cells rather, and injected it. The mouse was fine. But when he mixed the two together, I actually proved this by plating them out, like you do it. When he mixed non-encapsulated with the heat-killed encapsulated, all of a sudden, mice were dying. So he had somehow taken an element by killing the encapsulated bacteria. It did not destroy the element that would kill the mouse. And when he combined them with the ones that were not harmful, not only did it kill the mouse, but they started to grow capsules. So he changed them. He called it transformation. So I'll endeavor to show you how that works. I think. I hope. Should have loaded it before, but I didn't. So there's a zip file here. I have the animations separately if you need them. So take a look at where it is here. So we're going to look at bacterial transformation. I use the MP4s. I don't think the shockwave works.
DNA transformation involves transfer into a recipient cell. In the first step, double-stranded donor DNA binds to specific receptors on the surface of a competent cell. One strand of the donor DNA is degraded by nucleases while the other strand enters the cell. The single-stranded donor DNA pairs with an homologous region on the recipient DNA and is integrated into the recipient genome by a breakage and reunion mechanism called homologous recombination. If there are any differences between the nucleotide sequences of the donor and recipient DNAs, the mismatch repair system comes into play. The repair system removes either the donor or the recipient strand and replaces it with the complementary sequence. Since either strand may be repaired, some cells contain the new donor DNA and others have the original DNA sequences. In the laboratory, cells are plated on selective media so that only the transformants will grow.
So I'll run it by you to show you and really, if you understand this, you'll understand conjugation. Okay, we take it back without doing the audio. So this was a strand from a bacteria that was resistant to an antibiotic, ampicillin, very common. And so when that bacteria died, by whatever means they did so in the lab, they were able to observe that the naked DNA, and notice only one strand comes in. It's not, remember, it's double strand, only a single strand comes and the other one degrades. The first thing it does is it attaches not over here at the far end, but right where it came from the other bacteria. That's homologous recombination. It's almost like magically the DNA knows where to go. It has to do with the ends of the fragment. They fit in certain places. Now you have a mutated in red and a normal in blue. Mismatch repair is why it's 50-50 at best. Generally, we acknowledge it and we do experiments here. That's a standard experiment that the students have been doing for a long time. It's as likely to remove the mutated strand as it is the normal strand. Okay, so it's going to repair one or the other, so it's not reliable. It's a complex mechanism called restriction modification, which you don't have to know about. We utilize the enzymes that do the repair called restriction enzymes. That's how we're able to do DNA analysis like DNA fingerprinting, paternity testing. It's how we do crime scene DNA testing. We utilize these materials from bacteria. And what happens if we can prove, and this is what we do in the lab, we tag it and we actually put a fluorescent marker on it so that in the lab, we'll turn off the lights and shine ultraviolet light and like a bluish green fluorescence. That's how it is. Everything else, once it's there, DNA changes like this are permanent. It's going to keep, as long as nothing else happens to it, that DNA is a stable change that occurs. That's important as well. So that's the first one of these little bits of information that you need to be shared with. So what happens in transduction? Same darn thing, except a virus. The virus is the vector. The DNA is not just wandering around in the area and kind of makes contact at just the right place. We have a virus that's going to carry the DNA, so we call that a vector. So I'll show you the one with that, which is, and we're not going to do specialized, we're just going to do what's called generalized transduction. Specialized means in a particular area.
In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called a bacteriophage or phage. The phage attaches to the bacterial cell and injects its nucleic acid into the host cell. A phage enzyme is produced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and phage coat proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material.
So the only thing that's different is how it gets there. So this is your introduction to how viruses work. And a cold virus works this way in us. So what happens is you're susceptible to the virus. So it's a cold that's in your respiratory cells. In this case, the viruses don't look this way in the cold. they have a unique appearance, as you will see in our viral chapter, of bacteriophages, which are really oddball looking viruses. They look like from outer space. No joke. So here you can see in red is the viral. They're typically DNA viruses. They attach here. And obviously, in order for that cell has to be susceptible, meaning it has a receptor. So we're assuming that this bacteria is compatible with this virus, or liable to be infected. Very significant feature is that enclosure, which has a fancy name. It's called a capsid. Doesn't come in. It just shoots in the genetic material. In this case, the DNA. Once it enters, it immediately goes to work. First thing it does, it destroys the assembly of the host cell's DNA, fragments at the start. Then it's going to deteriorate. But while it's fragmented, now what viruses do is they start making not just one virus, but hundreds, if not thousands of viruses. Each virus has a certain amount it makes in a cell, and when it reaches, and a certain number of viruses like this one attain something called burst size. So let's say it's 500 of these. And what happens is that virus will reach its burst size, it'll cause the cell to explode and spit out 500 viruses. Those 500 will infect 500 more cells. So it's like n to the 500th power, not like n to the second power or 2 to the n power. It would be 500 to the n power. That's why viruses can clobber you and kill you in days. Somebody exposed to COVID, all of a sudden 2 days later, they're like, oh my God, I died. So what happens is it starts to, and what does a virus do when it assembles? It starts making a new closure and getting ready to be able to infect the next cell or group of cells. So it's making more DNA, and you see there that little sort of like key surrounding it. Inadvertently, viruses are there's nothing magical. Sometimes they surround, as you can see in the blue, some of the host cell's DNA. If that fragment happens to convey resistance or ability to make a toxin, which it frequently does, okay, now you have something that looks like a phage virus, but really isn't, because it doesn't have the phage DNA. Burst size is reached, it explodes out. The one with the original bacteria's DNA, with whatever it contains, then effectively attaches to and is injected in to the next cell in that population. And then everything else is transformation. Homologous recombination undergoes mismatch repair, whether it's included or not, and becomes stable and starts to replicate and replicate and replicate. The whole difference between transformation and transduction is the virus is a vector. And interestingly, this happens more frequently than you would think. You will see when we do the virus chapter how many bacteria have undergone a process called phage conversion, where it has imparted resistance and particularly toxicity. Why is Botox toxic? Okay. Why is diphtheria toxic? Because it's got a viral infection. And that viral infection is conveyed. So it's not an innocent process whatsoever. And those are the ones that are least likely to occur. So as we go back toward the PowerPoint and look at this, so the phage infects it, attaches, blah, 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 blah. Generalized transduction results when a fragment enters phage protein coat, and they call it, fancy name is transducing particle germ. So that's not a big deal. And they're just showing the same kind of illustration that you saw before by transduction. So there's not a lot else to say for now until we start to, in the next chapter we do, explore the bacteriophage virus a little bit more, which leads us to the most common by far, conjugation. Conjugation is the transfer of DNA between bacterial cells, typically in the same population, and they are contact. So it involves particular plasmids, those little circles of DNA, which typically are not attached to the bacterial's chromosome or nucleoid. Again, useful but non-essential. Okay, so they don't, and I would ignore the term replicons. In order to do this, those conjugative plasmids are sometimes called F, which stands for fertility plasmids. They do two things at a minimum. You can see them. I'll write them out. Those fertility plasmids could have anything on them, but at a minimum, they have two things. One, the ability to make the F pilus or fertility pilus, as you can see a little bit in that micrograph, basically a hollow tube that projects from the bacteria that's going to allow the DNA to move. And two, the ability to transfer the plasmid. It may have other properties like resistance or toxin manufacturing. Anything like that. It can impart any other property you might have. Make a capsule like you were seeing. So there's different things it could do, but those are optionals. Plasmids that have resistance are also called R plasmids. An F plasmid could also be an R or resistance plasmid, but may not. And a resistance plasmid doesn't have to be an F plasmid. They're not mutually exclusive, but you have to understand F plasmid is very, very specific. So that's the big player here, and you'll see the last animation that we're going to do today before we go, and I'm sure I'll go over it again and reiterate it. The term, wrong one. Here in the time we have left. So this is bacterial conjugation, I think. Hopefully this is the right one. Transfer the F plasmid. Conjugation and transfer of said F plasmid. Here we go.
Conjugation is a mechanism of gene transfer that requires direct contact between donor and recipient cells. F plus cells carry the F plasmid, enabling it to make an F pilus and act as a genetic donor. Recipient cells that lack an F plasmid are referred to as F minus. The F plasmid encodes for the F pilus, a protein appendage that attaches the donor to the recipient cell. The first step in plasmid transfer is contact between the donor and the recipient. The F pilus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell. The plasmid then becomes mobilized for transfer when a plasmid-encoded endonuclease cleaves one strand of the plasmid at a specific nucleotide sequence called the origin of transfer. A single strand of the F plasmid, beginning at the origin of transfer, enters the F minus cell. A complementary strand to the single-stranded plasmid remaining in the donor is synthesized via the rolling circle mechanism. Once inside the recipient cell, a complementary strand to the single-stranded DNA is synthesized. When F plus and F minus cells are mixed together, eventually all of the cells become F plus.
See, if we don't have enough time to discuss it, we'll continue the discussion with this on Friday. And like I said, I'll try to remember to, if not, if you don't see it today or tomorrow, remind me and I'll get a study guide out to you. Thank you very much.
Do you want our